Okay, so we're quite pleased to have with us Nish Reddy. He's a PhD, currently at MIT, but he did some of his training at UCSF at the Cell Design Institute. And I'm just going to, you can see here the title of his talk, Engineering Immune Suppressor Cells, Designing Cell Programs to Treat Inflammatory Diseases. A little about him, his bio. He completed his undergraduate training at UC Berkeley in bioengineering with a focus on systems and synthetic biology. He recently completed his doctoral thesis in Wendell Lim's lab at UCSF Cell Design Institute, working on immune cell engineering to design smarter cell therapies. And currently he is a postdoc fellow at MIT. His recent paper, which I'm going to pop into the chat, is Engineering Synthetic Suppressor T Cells that Execute Locally Targeted Immunoprotective Programs, that was recently published in Science, and so in December 2024. So adding people in here. I'll drop that into the chat, and I look forward to hearing more about your work, Nish. Thanks for joining us.
Yeah, thanks, Monica, for having me here. I'm excited to share some of the work we've been doing over the last several years on engineering immune suppressor cells. And so this was work that I did in Wendell Lim's lab at the UCSF Cell Design Institute, and it was a very close collaboration with Audrey Parent's lab and Matthias Hebruck's lab. And so at the Cell Design Institute, we're really interested in understanding how cells work. And we know cells have this remarkable ability to make really complex decisions by using molecular signals between other cells and their environment. For example, a T cell that recognizes a cancer cell has really complex machinery that allows it to specifically recognize particular cells and not other cells. And these cells also have the ability to make really precise responses, like, for example, killing a cancer cell. And so one way to study these cellular processes is by deconstructing how these cells are functioning in their native context. But what we do at the Cell Design Institute is try to understand how these cells work by constructing them from the bottom up. And this is called synthetic biology. And what we're interested in doing with synthetic biology is both to explore the ways that evolution has wired different cellular processes, but also to explore alternate wirings of these cellular processes. And this is really important in understanding more fundamental rules for how cells function. And these design rules can be really important in tailoring particular functions for therapeutic settings. And so we know that cells have several advantages over these traditional modes of therapies like small molecules and biologics, because cells can actually act locally. So they can detect particular signals in their microenvironment or other cells and then execute a particular response. And so because of this, cell-based therapies have the potential to treat many challenging diseases. And for example, in the type 1 diabetes context, being able to protect a transplant from rejection or protect islets from immune attack. And so the archetypal example of cell therapies that many of you might be familiar with are CAR T cells. And so CAR T cells against cancer have been FDA approved and have been advancing in the clinic. And these cell therapies rely on taking blood samples from a patient and then transducing these cells, these T cells, with a receptor, a CAR receptor, that basically programs these T cells to attack and kill a cancer. And so these CAR T cells rely on a native T cell activation pathway to activate the T cell and promote this cytotoxic activity. But several years ago in the Lim lab, several people have developed this synthetic Notch receptor. And how this synthetic Notch receptor works is it has an extracellular recognition domain, like a CAR, that allows it to detect a particular signal in the body. But rather than activate a native T cell program, it can activate a custom therapeutic response. And so these synthetic Notch receptors are highly customizable in that their recognition domain can be changed to reprogram these cells to detect different targets. And it has a transmembrane notch core that, when cleaved upon binding, can release a transcription factor and induce the production of a custom transgene. So in short, these synthetic receptors allow you to have a cell recognize a particular target and induce a custom response. So this video just illustrates that process. So these synthetic notch receptors have a recognition domain, and then upon binding to a particular target molecule on another cell, cleavage of this transcription factor allows you to induce the production of a custom output. And as I mentioned, these receptors are highly modular, and so they can be reprogrammed by changing out their extracellular recognition domains or changing different intracellular response elements. So one thing that we were really focused on was this question of, can we engineer an immune cell that is capable of targeted immune suppression? And this is important because our immune system requires this really complex balance between inflammatory and tolerogenic signals within each tissue. And we know that these anti-cancer CAR T cells, for example, are able to locally promote targeted inflammatory responses. But can we engineer a cell that can do the opposite, that can have targeted suppression activity? And so there's a variety of different approaches to do this. So one way would be to redirect native immune suppressor cells like regulatory T cells or myeloid suppressor cells and have them go to sites of infection or inflammation. But we wanted to see if we can, rather than take a cell that already is able to induce immune suppression, have a cell type like CD4 T cells that normally doesn't have immunosuppressive capabilities and introduce those using synthetic circuits. And so if we use a cell like a CD4 T cell, these cells are highly engineerable and easy to acquire, and we can transduce these cells with SynNotch circuits that can induce custom payloads in an antigen-triggered manner. And so to show initial proof of principle, we engineered these CD4 T cells with a SynNotch circuit that recognizes a model antigen, CD19, and induces the production of these custom suppressive payloads. And so we explored a diverse set of different immunosuppressive agents that we know from the literature, such as suppressive cytokines or inflammatory cytokine sinks, inhibitory receptors, or also signals that promote proliferation of cells. And we wanted to see, would any of these payloads induced by these engineered suppressor cells act locally to suppress immune responses, such as a cytotoxic T cell. And so we created these 3-cell co-culture assays where we had a target cell that expressed 2 antigens, HER2 and CD19, where our suppressor cells recognized CD19 on these target cells. And a second cell type, CAR T cell, which is modeling a cytotoxic T cell response, recognizes HER2 on these target cells. So we wanted to see which of these engineered suppressor cells, which we call synthetic suppressor cells, are capable of inhibiting CAR T cell proliferation and killing. So we looked at both the ability of these suppressor cells to block CD4 and CD8 CAR T cell expansion. And what we see is that certain payloads like IL-10, TGF-beta, and PD-L1 are capable of blocking CD4 CAR T cell expansion, whereas payloads like TGF-beta and PD-L1 are able to block CD8 T cell expansion. And also interestingly, we see IL-10 has specific activity against CD4 T cells, but not against CD8 T cells. So there's some specificity in what cell types these inhibitory signals are able to block. So we see that many of these payloads are able to induce some suppression of CAR T cell activity, but we wanted to see what would happen if we combine payloads together. So we hypothesized that combining payloads together would induce much stronger suppression activity. So we see that combinations of inhibitory signals like TGF-beta, IL-10, PD-L1 with CD25, which is a sink for the proliferative signal IL-2 that T cells require to grow, actually works synergistically to suppress CAR T cell expansion and improve target cell expansion. But now we did the same thing with CD8 T cells, and we also see that specifically TGF-beta and CD25 are able to suppress CAR T cell proliferation, which is on the x-axis, and then target cell survival is increased on the y-axis.
Can I just interrupt you for 1 second, Nish? How local is this? Is this a global or a local effect? Is this just happening right at the site of some kind of T cell approach, or is it a systemic?
Yeah, sorry. So just to clarify, this is all initially we did all these in vitro. So these are just 3 cells mixed together in cell culture. And I'll show you some work later on what we did in vivo.
Great, got it. Thank you.
Yeah, so in vitro, we sort of explored this whole space of the combinations of these suppressive payloads, both to suppress CD4 and CD8 cytotoxic T cell activity, and find that these combinations of payloads drives stronger suppression. And so if we look at this in our cell culture over time, and we look at CAR T cell expansion on the y-axis, what we see is that in the absence of any suppressor cells, these CAR T cells are able to expand several fold over the course of 9 days. But now when we introduce into this co-culture these suppressor cells that are inducing individual payload combinations, we see that the CAR T cell expansion is significantly reduced, and that this combination of payloads drives more effective suppression of CAR T cell expansion and killing. And then similarly with the survival of these target cells that are normally killed by the CAR T cells, we see that these combinations of payloads are able to protect these target cells from T cell killing. So we can also look at this more quantitatively, looking at different doses of suppressor cells. So on this x-axis, we have the initial number of suppressor cells that we add, and on the y-axis is the level of suppression of this CAR T cell expansion. And what we see is that at lower doses with this combination of payloads, we see much stronger suppression of CAR T cell expansion compared to each individual payload alone. So interestingly, these two factors, CD25 and TGF-beta, are actually features of natural regulatory T cells. And so our synthetic suppressor cells actually mimic some of these properties, as Tregs are known to have sinks for this pro-inflammatory cytokine IL-2, and also act as a source for these inhibitory cytokines. But we show that these two signals in these minimal circuits are still capable of inducing local immune suppression. So when we engineer our CD4 T cells to express CD25, what we notice is actually 2 things. So CD25 is often thought to act as an IL-2 sink. So in line with that, we see that in this in vitro cell culture over time that has exogenous IL-2 added, that cells that express CD25 deplete IL-2 more effectively from the media than control cells that don't have CD25. But secondly, we see that these engineered suppressor cells that have CD25 actually proliferate more. So these IL-2 sinks have 2 roles, both acting in the consumption of IL-2, but also enhancing proliferation of the suppressor cells. And when we look at cells, the suppressor cells and the killer cells in co-culture, what we find is that these suppressor cells that are expressing CD25 actually have stronger IL-2 signaling. So they have higher levels of CD25, and as a result have higher levels of phospho-STAT5, which is a marker of IL-2 signaling. So to test this hypothesis, what we did was we separated out these suppressor cells such that CD25 and TGF-beta are not present in the same cell, but rather they're expressed by 2 different cells. So do these 2 separate cells act just as well? And what we find is that that's not true. So the suppressor cells that are producing both TGF beta and CD25 are much more effective at suppressing CAR T cell killing than separating these out into two different cells. And this is in line with what we observe with the function of CD25, in that CD25 seems to promote the expansion of these suppressor cells, and that is really critical for this synergistic effect. So in summary, from this in vitro reconstitution, what we find is that engineered suppressor cells that produce an inhibitory signal like TGF beta and also produce CD25, which is a sink for IL-2 that also acts in promoting suppressor cell expansion, and that these signals can act synergistically to inhibit CAR T cell activity. And so the combination of these factors leads to more IL-2 depletion in this in vitro cell culture, but also more TGF beta secretion as a consequence of suppressor cell expansion. So these suppressor cells are basically expanding in response to the IL-2 signal, and as a result, producing more TGF beta, which sort of acts synergistically to drive CAR T cell suppression. So we demonstrate that these engineered cells are capable of suppressing T cells in vitro, but now we wanted to look in vivo and whether these engineered cells can act locally to suppress immune responses. And to do this, we started out with a model where we have a mouse that is transplanted with two different tumors. So each flank has a tumor that contains the antigen HER2, and one flank has this tumor also express CD19. So HER2 is the antigen that the CAR T cells are recognizing. So both tumors are under attack by the CAR T cells, and these suppressor cells are only able to recognize CD19, which is on the dual antigen tumor. And so we wanted to see whether these suppressor cells in vivo are able to protect this dual antigen tumor from attack, and whether they could do this without affecting the single antigen tumor. And so what we see is that in the absence of any suppressor cells, when we just have CAR T cells alone, that both tumors are cleared at pretty equal rates. And now if we have suppressor cells that are producing one of these suppressive payloads like CD25 or TGF beta, that those aren't sufficient to protect either of the tumors. But now when we have suppressor cells that are producing both CD25 and TGF beta, we see protection of the dual antigen tumor while the single antigen tumor is still cleared. So this suggests that these synthetic suppressor cells are able to locally inhibit CAR T cells in vivo. So we wanted to look at whether these synthetic suppressor cells could locally block immune responses in a couple different contexts. So one of the contexts that we were interested in was in blocking the toxicities of CAR T cells. So there's a lot of CAR T cells that are used for trying to treat solid tumors, but many of the antigens that these CAR T cells recognize are also found on other tissues. So can we engineer suppressor cells that recognize this cross-reactive tissue and suppress CAR T cell activity there? So these are often called NOT gates, and there's a lot of interest in engineering CAR T cells with this sort of more Boolean tumor recognition, where you can recognize tumor but not have this off-target toxicity. So we compared our engineered suppressor cells to other strategies for designing these NOT gate circuits. So one strategy to engineer these NOT gates is to express both a CAR that programs these T cells to kill a tumor, but also an inhibitory CAR that recognizes another antigen that would inhibit within the same cell the pathway that activates this CAR receptor. And we compared this to our strategy of using both a CAR T cell as well as a second cell, a suppressor cell that recognizes the cross-reactive tissue. And so what we see is that in the context of this two tumor model that I mentioned before, that this ICAR receptor is not able to completely suppress killing of this dual antigen tumor. Whereas our two-cell system that has both a CAR T cell and a suppressor cell leads to more robust protection of this dual antigen tumor from CAR T cell killing. So the other context that we were really interested in, and the one that's more relevant to the type 1 diabetes community, is protecting islet transplants from rejection. And so this was a close collaboration with Matthias Hebruck and Audrey Parent's labs at the UCSF Diabetes Center. And over the last several years, they've been using human pluripotent stem cell derived islets as a replacement for islets in type 1 diabetes, and have been developing these protocols to design these enriched beta cell clusters. And so working with them, we've generated these beta cell clusters and introduced our model antigen CD19 into them by lentiviral transduction. And so we can model cytotoxic T cell killing of these islets by using a CAR T cell that recognizes HLA-A2 on these beta cell clusters. And so these CAR T cells in vitro are able to kill these beta cell clusters within 3 days in the absence of any suppressor cells. And what we find when we introduce our synthetic suppressor cells that are producing both TGF-beta and CD25 is that we have better protection of these islets from T cell killing. So one interesting thing that we observed during this in vitro suppression assay with the beta cell clusters is that in the absence of any suppressor cells, these CAR T cells form these massive aggregates of T cells on the periphery of the beta cell clusters. And so these aggregates kind of build until the beta cell clusters are eventually cleared. But in the presence of these suppressor cells, especially suppressor cells that are producing CD25, we see that individual CAR T cells that are in contact with the beta cell clusters actually become surrounded by these suppressor cells, almost like a detergent that's kind of preventing the CAR T cells from expanding and eventually killing the clusters. And so we see some of the similarities here to what we see with regulatory T cells in secondary lymphoid tissues, where individual CD4 T cells that are being activated by an APC actually get surrounded by T cells to prevent, self-antigen recognition or these sort of undesired immune responses. So we demonstrated that these engineered suppressor cells are capable of protecting these EBC in vitro, but now we wanted to see whether they can protect these when they're transplanted in vivo into a mouse. And to do this, we had our EBCs transplanted under the kidney capsule of these NSG mice, and we modeled T cell killing of these transplants by using that anti-HLA-A2 CAR T cell. And so we can look at whether these suppressor cells are able to improve the survival of these transplants. And so what we see is in the absence of any T cells that these transplants measured here by bioluminescence imaging remain pretty stable for long periods of time. And we've measured up to 6 weeks, and these transplants remain stable in these immunodeficient mice. But now when we introduce these CAR T cells, the transplants are cleared within about a week. When we have our CAR T cells and suppressor cells, we see that these transplants remain protected or much better than with the CAR T cells alone. And so this is histology images of anti-CD19 staining, showing that these transplants are remaining intact in the presence of suppressor cells. So we wanted to test whether this protection was antigen specific. And so what we did was we engineered EBCs that both had the CD19 antigen and EBCs without the CD19 antigen. And we find that in the absence of the CD19 antigen, suppressor cells are not able to function. And we see that both transplants in the presence or absence of suppressor cells are cleared at equal rates. But with the CD19 antigen, we see protection of these transplants with the suppressor cells. So this suggests that these suppressor cells are able to drive antigen-specific suppression. So we wanted to see whether these transplanted EBCs were still functional after protection. And to do this, we did a glucose challenge test where we injected fasted mice with glucose and measured the human C-peptide that's produced by these under fasting conditions and 30 minutes after glucose injection. And what we see is that in the presence of CAR T cells, that we almost completely lose the ability to produce C-peptide, whereas at similar levels to the no T cell control.
Oh, I'm sorry, Nish, but you just still froze just for a second there. I don't know if you want to just reiterate that last point.
Oh, sorry. So basically we did a glucose challenge test to assess whether these transplants were functional after immunoprotection by these suppressor cells. And we found that with CAR T cells alone, that these transplants were, these transplants were cleared in the presence of CAR T cells alone, but now when we add our suppressor cells, that these transplants are protected from immune rejection and also remain functional in their C-peptide production.
Great, got it.
So what we demonstrated here was an initial proof of principle that we can engineer synthetic suppressor cells that are capable of blocking cytotoxic T cell responses. And we show this in the context of tumor knock-gate killing circuits, as well as in protecting islet transplants from rejection. And these designer suppressor cells could be tailored for many different types of autoimmune and inflammatory disorders, including protecting native tissues like native islets from immune rejection. So in summary, what we learned from the synthetic reconstitution is that minimal circuits, specifically circuits that use these inhibitory signals like TGF-beta or IL-10 with CD25, a sink for IL-2, are able to suppress cytotoxic T cell killing. And that these engineered cells are capable of locally targeted immune suppression in vivo in these contexts. So additional work in future is needed in order to understand how these cells function in different disease contexts and in local tissue microenvironments. But what we demonstrate here is the proof of principle that these cells are able to exert immune suppression and that they can act locally. And so this paper was published last month in Science, and the plasmids are now all available on Adgene. So yeah, feel free to check out the paper.
Great. So are you continuing this work at MIT?
I'm not, but people at the Cell Design Institute and Wendell Lim's lab and Audrey and others are still continuing this work.
Great. Just sort of like harking back, sort of what challenges do you or they anticipate in translating this technology from mouse models to humans?
Yeah, so I think there's several kind of challenges. So like one is understanding how these cells are able to function in different disease like for example in a type 1 diabetes model or transplant model. How effective are these cells at suppressing these more complex immune responses? So we demonstrate here that they can suppress cytotoxic T cells, but what other signals may be needed to suppress other cells. I think one key advantage of these types of systems compared to trying to isolate some of these rare suppressor cell populations and engineer them is that we're using a cell type with CD4 T cells that is very easy to engineer, but may potentially they may be needed to be dosed multiple times over the course of transplant or autoimmune disease. and what is the lifespan of these cells, in a
Yeah.
mouse or patient. I think those are kind of some of the major questions that remain.
Yeah, well, for instance, like Bio just had a win with their one patient, with their implant, with their the implant lasted 4 weeks. So, when you're thinking about these kinds of this kind of cell therapy, like would you want, how long would it last, and how might you replenish, replace, all those kinds of questions are kind of floating around.
Yeah, and I think like something like this could be a complementary approach to what Sana's doing with engineering the transplant itself.
Yeah, it seems
Additional,
like it could be a very nice synergy.
yeah.
Um, I wanted to ask you, what potential off-target effects or like any safety concerns would you anticipate with these types of T cells?
Yeah, so I mean, one of the things is whether these CD4 T cells have natural, reactivity through their TCRs, and so, in the future there could be interest in, knocking out these TCRs and reducing some of that toxicity, or in some of the factors that we produce, like TGF beta, obviously TGF beta is known to have these effects on fibrosis and other things that may be of a concern. What we do find is that when these factors are produced through SynNotch, that they can act locally, and so some of these potential systemic toxicities are mitigated in that way. But
Yeah,
we also know that we can deactivate SynNotch using pharmacological inhibition. So in those sort of cases, this could also be a way to control those toxicities.
got it. Are you aware of any companies right now that are scaling this tech, that could be sort of repurposed for the type 1 diabetes space?
Yeah, so I think several groups are, including the Cell Design Institute, are doing trials now to test SynNotch, mainly in the cancer context right now. But I think
Right.
proof of principle in that context will kind of accelerate translation of this into other immune disorders and type 1 diabetes especially.
Are there any, you kind of imagine there might be some stealth biotech, but are there any other biotech that you know of and that you can speak to that are doing this?
Um, not not yet. I think, yeah.
All right, we'll circle back. Well, it was really interesting. Fingers crossed that Audrey Parent and Matthias Hebrock can, bring this to bear in the type 1 diabetes field. And, perhaps there would be as we sort of touched on, a symbiosis with a company like Sana to really elevate what they've got going. So,
Yeah,
yeah,
definitely.
sounds
Yeah.
good. Oh, sorry.
No, I'm just saying it'll be really exciting if this work can progress and then reach translation and help patients eventually. So we're working
Yeah.
for.
Sounds good. Thanks for your role in bringing this to, to publication and for speaking with us today. Hope you have a great rest of your day, Nish.
Thanks, Monica. Thanks everyone.
Yep.
Bye.
Bye-bye.